Assalamualaikum students. Before going to our educational topic, please subscribe my channel Muntazar Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. May Allah bless you its reward. Today our educational topic is DNA as a genetic material. In our this lecture we will conclude that how does DNA perform functions as a genetic material. We will approve the DNA as a genetic material. For this question we can say that biologists of 20th century apprehended that the genetic material must have some assertive characteristics. These characteristics explain the properties of life. For this purpose, the first characteristic is control over protein synthesis. First point is that the genetic material must be able to code for sequence of amino acid in proteins. As we know that the amino acids are building blocks of polypeptide chain or protein. So the genetic material must supervise the translation process. Second important characteristic of the genetic material is replication. So the genetic material must undergo replication process before cell division. During replication, the genetic material synthesizes another copy of itself. Copy means replica. So it is necessary for the genetic material to synthesize another replica of itself. This replication process takes place before cell division during S phase of cell cycle. When parental cell undergoes cell division and forms two daughter cells. Both daughter cells receive single copy of genetic material that is already replicated in the parental cell. The third characteristic that is possessed by the genetic material is located in nucleus. The genetic material never present in either side of a cell except nucleus. Nucleus is the main and central organelle of a cell that contains genetic material of eukaryotic cells. Its fourth characteristic is mutation, meaning that the genetic material must be able to change over time for evolutionary changes, for changing the life being and its existence, to adapt the better adaptations according to the environment. The genetic material must be able to mutate by carrying mutations in the DNA to chromosomes and chromosomes to genes, and finally from genes to the protein. So all of these expectations are fulfilled by a single molecule, that is deoxyribonucleic acid, shortly called DNA. So DNA is the only molecule that fulfills all these requirements, such as it has control over protein synthesis, undergoes replication process as it synthesizes its own copy, is located in the nucleus and undergoes mutation. It is important point to note that the DNA molecule belongs to the nucleic acid. A cell contains two types of nucleic acid. The first one is deoxyribonucleic acid, shortly called DNA, that transfers genetic information from one generation to the next generation and acts as a genetic material. The second type of nucleic acid is ribonucleic acid, shortly called RNA. The RNA may be a transfer RNA, a ribosomal RNA, and a messenger RNA. These are the three types of RNA. All these three types of RNAs are produced in the nucleus of the cell and then moves to the cytoplasm. In the cytoplasm, these RNAs engaged in protein synthesis. So we can say that RNA molecule, according to the instruction of DNA, participate in protein synthesis process. Both contain similar building blocks that are called nucleotides. Next, we shall study about basic structure of DNA and RNA. In 1920s, the basic structure of nucleic acids that include DNA and RNA was derived by biochemist P.A. Levin, who originated that DNA molecule contains nucleotides. These nucleotides contain three main components. These three main components are as follows. The first one is phosphate group. Its chemical form is PO4, like this. The second component is 5-carbon sugar, which has a form of a pentose sugar, like this pentose sugar. The word pent means 5. As it has 5 corners, so it is known as a 5-carbon sugar or a pentose sugar. third component of the nucleotide unit is nitrogenous bases. When all the three components combine, then they form a nucleotide. Now we see that how these three components combine in a nucleotide. As we can see that in a DNA nucleotide, the central position is occupied by a pentose sugar. At the right side is present a nitrogenous base, whereas at the left side is present a phosphate group. Now we shall discuss all the three components in detail. Firstly, we shall discuss about phosphate group. A phosphate group is present in this form, but when this phosphate group removes one hydrogen, then it attains a negative charge at its associated oxygen, like this. Similarly, by removing further hydrogens, it attains increased negative charge. And finally, a time comes when all the three hydrogens are removed and phosphate contains only negative charges. At that time, it attains this form, in which this phosphate group contains only negative charges. Next, we shall see the pentose sugar. These are the pentose sugars, one for DNA and other for RNA nucleotide. Pentose sugar present in DNA nucleotide is different from the pentose sugar that is present in RNA nucleotide. Now you are thinking that why DNA and RNA nucleotides are contained different pentose sugars. What is the major difference in between the both sugars? But before understanding the answering of these questions, we must consider the numbering of pentose sugar. Now this is a pentose sugar. It attains the carbon numbers as follows. At the uppermost corner, there is present oxygen. At the first right corner of oxygen, there is present carbon number 1. At the second right corner of oxygen, there is present second carbon, whereas third carbon is present here and fourth carbon is present at the first left corner to oxygen. Fifth carbon is located just outside the ring. This is the simplified structure of pentose sugar. Its first carbon contains hydrogen and hydroxyl group. The difference is present at the second carbon, which we will discuss later. So the third carbon contains a hydroxyl group and a hydrogen. Fourth carbon contains a hydrogen atom and also attached to the fifth carbon. This fifth carbon contains two hydrogen atoms and one hydroxyl group. The most important carbon of these five carbons is the carbon number 2, which determines whether the sugar is ribose or deoxyribose. In the case of ribose sugar, that is present in RNA, carbon number 2 contains two functional groups, that are hydrogen and hydroxyl group. So in RNA, second carbon contains hydroxyl group, whereas in the case of DNA, oxygen from the second carbon is removed and this second carbon has two attached functional groups, that are only hydrogens, two hydrogens. So this is the major difference between a deoxyribose sugar that is present in DNA and a ribose sugar is present in RNA. RNA contains at its second carbon a hydroxyl group, whereas in the case of DNA molecule that contains deoxyribose sugar contains only a hydrogen instead of hydroxyl group. So the presence of hydrogen or hydroxyl group determines that the sugar is ribose sugar or a deoxyribose sugar. The next component of the nucleotide is nitrogenous bases. These nitrogenous bases are of two types. The first one is purines and the second are pyrimidine. Purines are double ringed and larger bases, whereas pyrimidines are single ringed and smaller bases. Purines include adenine and guanine. As we can see that adenine and guanine both are double ringed and larger bases, whereas pyrimidines are uracil, cytosine and thymine that are single ringed and smaller bases. Next, we shall study about the attachment of pentose sugar to the phosphate group and nitrogenous bases. Let's see which carbons of the pentose sugar are attached to these phosphate group and nitrogenous bases. The biochemist P.A. Levin derived that DNA and RNA molecules are synthesized by repeating units that are called nucleotide. This nucleotide nitrogenous base is attached to the carbon number 1 of pentose sugar. The carbon number is not affected by the presence of a ribose sugar or a deoxyribose sugar. If the sugar is deoxyribose or ribose, nitrogenous base is always attached to the carbon 1 of pentose sugar. The nitrogenous base is thymine. Carbon 1 is always attached to the N1 of thymine. The similar case is for cytosine and uracil in the case of RNA nucleotide. But if there is present a double ring base adenine, then carbon number 1 of pentose sugar will be attached to the nitrogen 9 of double ringed adenine. The same rule is applied with the guanine nitrogenous base. It is also a double ringed nitrogenous base. It will also be attached by its nitrogen number 9 to the carbon number 1 of pentose sugar. Next, we will see the phosphate group, that at which carbon of pentose sugar phosphate group will attach. This diagram we can prove that phosphate group is attached to the carbon number 5 of the sugar. In addition, a free hydroxyl group is also attached to the third carbon of this sugar. So we will understand the dehydration synthesis. In this reaction, hydroxyl group of one nucleotide and phosphate group of the second nucleotide. So this is a dehydration synthesis reaction during which a water molecule is eliminated and a covalent bond is formed between the two nucleotides. One nucleotide is present at the upper and the other is present at the lower side. So the phosphate group that is present at the 5 carbon of pentose sugar react chemically with the hydroxyl group present at the third carbon of sugar. Like this to form long chains of nucleotides of DNA and RNA. In other words, we can say that 5-phosphate group of one nucleotide and third hydroxyl group of second nucleotide. Both these nucleotides react chemically with each other to form long chains of nucleotides of DNA and RNA. Like this central covalent bond, which proves that when hydroxyl and phosphate groups react chemically with each other, then they form polynucleotide chain. This 5-phosphate group is always available to bind with the hydroxyl group of an other nucleotide, whereas lower hydroxyl group is also available to bind with the phosphate group of an other nucleotide to form polynucleotide chains. In this polynucleotide chain, phosphate group is linked to the two sugars. First is here and the second is here by means of a pair of ester bond. First pair is this, that is POC, a pair of ester bond, whereas the second ester bond is here, that is also POC. So by means of a pair of ester bond, two sugars that are sugars of nucleotides, these two sugars are bonding to the phosphate group. This linkage is called phosphodiester bond, as it links two nucleotides by means of a phosphate group. In this way, many thousands of nucleotides join together, form long chains. Linear strands of DNA or RNA almost always have a free 5-phosphate group at one end and a free third hydroxyl group at the other end. These 5-phosphate group and third hydroxyl group always available for the attachment of a huge amount of nucleotides to form very long chains of DNA or RNA. Then these DNAs or RNAs chains aggregate to form double helical DNA strand or RNAs. So this was an introductory video of nucleic acid, that is explaining the basic structure of nucleic acid including DNA and RNA. In our subsequent videos, we will discuss about mutations, recombinant DNA technology, respiration, glycolysis, Krebs cycle and many other topics. So make sure to subscribe our channel for further upcoming videos, and also make sure to like my videos at YouTube channel Muntazir Mehdi Science TV for receiving the notification